Hello guys, let's go ahead and jump right into chapter 10, section 3, gene linkage and polyploidy. Essential questions: how does the process of meiosis produce genetic recombination? How can gene linkage be used to create chromosome maps? And why is polyploidy important to the field of agriculture? 3 vocabulary terms, one of which is a review. Okay, so hopefully you've written those 3 down somewhere in your notebook. So let's talk about genetic recombination. The new combination of genes produced by crossing over an independent assortment is called genetic recombination. I like to think of it like shuffling a deck of cards. Okay, so it mixes everything up so you don't get all blue cards in Uno. Okay, you want genetic recombination. You want to have a mixture of genes. Combinations of genes due to independent assortment can be calculated using the formula 2n, or 2 to the nth power, where n is the number of chromosome pairs. So if we talk about humans, we have 23 pairs of chromosomes, so it would be 2 to the 23rd power. Any possible male gamete can fertilize any possible female gamete. Okay, so that means that the possible combinations after fertilization happens, after that sperm and egg fuse, is 2 to the nth times 2 to the nth. So if we're talking about humans, that would be 2 to the 23rd power times 2 to the 23rd power. So just think about how huge that number is. There's lots of possible combinations of genes. Gene linkage. Genes located close to each other on the same chromosome are said to be linked. That means that they are associated with each other. They're not attached. They're really, really close. They're linked. They usually travel together during gamete formation. That's why you see a lot of people with blonde hair and light eyes, because those genes are linked. Okay, they're not the same gene. They're not attached to each other. They're just really, really close. Gene linkage results in an exception to Mendel's law of independent assortment. They can be separated, but not very often. Okay, if genes are closer together, they're likely to travel together. All right, a chromosome map. It shows where chromosomes or where genes are located on a chromosome. And we can determine this by using crossing over data. Crossing over occurs more frequently between genes that are farther apart. So if genes are further apart, we know that they are more likely to get swapped during crossing over. So crossover data can be used to create chromosome maps depicting how genes are arranged on a chromosome. If we look at this diagram, it's more likely that V will not travel with Y because it's further apart. Okay, W will go with either V or Y during crossing over. Okay, so the closer genes are on the chromosome, the less likely they are to be separated. Polyploidy is the occurrence of one or more extra sets of chromosomes in an organism. A triploid organism is designated 3N, which means that it has 3 complete sets of chromosomes. Many agricultural crops are polyploid. Wheat has 6 copies of every chromosome. Oats also have 6, and sugarcane has 8. Okay, strawberries have extra pairs. I don't remember what number, but they have a lot. Polyploid plants often have increased vigor and size. That means that they're stronger. They're more likely to reproduce. They're more likely to have lots of offspring, and they're probably going to be larger in size. Okay, that's why they have been able to survive all of these years through evolution, because they have those extra sets of chromosomes. They are more vigorous. They're more suited for their environment. And that's it, guys. If you have any questions, let me know.